If we return to the Hill equation, okay, where fractional saturation was given by the ligand concentration raised to n, the number of ligands that can attach, you can rearrange this and solve for ligand concentration raised to the n. Right, so move the bottom over. I don't know what's happening here. Let's just get to the good part. Here we go. Okay. So we're going to rearrange our Hill equation so that we can get ourselves into our log-log units, or our plot here. And so if we take the log of both sides, again, what we'll get is this log of fractional saturation over 1 minus fractional saturation. And then we have n times the log of the ligand concentration minus the log of Kd. We have Hill slope. Again, like I said, n will give you the slope. When we first introduced this plot, we said if it has anything but a slope of 1, we know that we have more ligands involved, something's going on besides a 1-to-1 binding. And so now, again, if you saw a plot like this, you could take the slope and tell me how many ligands bind to that particular protein. If we fit our binding isotherm in the region around, like I said, F equals 0.5, you get the Hill coefficient. So then the slope of this is our sort of n sub h. The theoretical max of any Hill coefficient is equal to the number of subunits that the oligomer has. Okay, so again, if it binds one ligand in each of its subunits. So the theoretical max for hemoglobin is 4. Okay? This is the reality. This is the real curve for hemoglobin. It has a Hill coefficient between like 2.5 and 3.5 is sort of the real answer. We're not, sort of actual data fitting of this is outside the scope of this class. But the way you would get this is you could actually run a series of binding assays and plot them. And as you change concentration, you would be able to fit your results to this. And what you'll see is just, it's impossible to reach your theoretical sort of maximum. Okay. So here's how the curve looks different if you have negative cooperativity. Instead of a slope that is higher than 1, instead you'll have a slope that would be shallower than 1, right? So in this case here, we had sort of slope of 1 for each of our 2 different sort of states, but then we know we have a slope greater than 1 if we see positive cooperativity, whereas if Kd of binding to the first site lowers the affinity for the second site. Did it switch? Okay. I don't know. It left the colors the same, which is maybe a poor way to do it, because in this case now, these are sort of the opposite. Because once the first one binds, the conformational change, rather than being like a favored state, is the second binding site is now having the higher Kd, the lowered affinity. But again, when you fit the slope here, you'll now have something less than 1. So again, you could have a curve just for R state, just for T state, and then your sigmoid would fit in between. 2 more quick models. I won't ask detailed questions about this. So this concerted model is an equilibrium description. So you could write a set of equations for like describing the occupancy of every single state. And as each one adds onto it, you could have separate equilibrium constants, Kt and Kr, which would be Kds for the 2 different states. C here is a ratio of these 2 constants. L here is given by the concentration of the T state versus the concentration of the R state. The coefficients are the multiplicities. I won't ask you any questions about this. What I do want to point out is that in 2013, an article came out, the 50th anniversary article of the ubiquity and utility of the NWC model. And here is a quote from that paper. With the use of statistical mechanics, the mathematical implementation of this model links problems that seem otherwise to have no ostensible biological connection, including ligand receptor binding, ligand-gated ion channels, chemotaxis, chromatin structure, gene regulation. This model is used for pretty much everything. It's very, very valuable. And here's a figure from that. And in the end, what it's giving you is that the probability of T versus R state is determined by the energy of the state and partition function. And these equations, if you could read them, are essentially the same equations we saw for calculating partition function. Okay? So I invite you to take a look at that paper. Okay? Sequential model is another improvement. This is one where they flip the shapes of the square and the circle such that T is now the circle and R is now the square, whatever. But this is an improvement over the concerted model because, again, what we said with the concerted model is that once the first ligand binds, everything changes its conformation at once. And so now it's easier for all the additional ones to bind. It's a great model. It's pretty impractical in real life. So like I said, one of the problems is you can only use it for systems with 1, positive cooperativity, and 2, you would have to have like perfect symmetry of all your subunits. So it's sort of not ideal. This sequential model or the KNF model assumes progressive switching from T to R state. And so that's sort of what's illustrated here. You still have separate sort of binding constants or dissociation constants for each of these. But the first state changes, then when the next one binds, the next state changes. And in this case, each of these changes, you can then have negative cooperativity. In this case, you could also have, like I said, there's just a couple different things that could go on. But other than in like a very broad conceptual sense, like sequential is a better model than NWC because it doesn't have to assume positive and it doesn't have to assume all of the conformations change simultaneously. Those are sort of the broad questions I would ask you. Sections 16 to 22 of Chapter 14 cover more structural details of the model and sort of evolutionary conservation of allostery with respect. All of the discussion is all in the context of hemoglobin. I won't ask you specifics about those, but I do think it might be worth reading. Like I said, same thing with like the bacterial chemotaxis example. If you want to get yourself familiarized with an ultrasensitive system, and if you understand how that works, then a more simple one that I give you on the exam, you might feel more comfortable accessing it for the first time. Next time, we probably will do little, if no, review so that we can get through as much of Chapter 15 as we possibly can. We'll be talking about reaction rates and rate kinetics. I think that's